In this lab, I want to talk about the lymphatic system histology. But before I start talking about this part, I want to show you the lymphatic organs. As you see in this picture, we have lymphatic organs in different parts of our body. In this picture, you can see the lymphatic vessels, and the function of lymphatic vessels are collection of the lymph and bringing back to our blood circulation. You can see two primary lymphatic structures like the bone marrow and the thymus here. They are responsible for maturation of lymphocytes. The bone marrow can mature B lymphocytes and the thymus can mature T lymphocytes for us. We have a spleen in the left upper quadrant of our abdominal cavity. The function of the spleen is filtration of the blood, and also it can store blood, platelets, and red blood cells for us. You can see the lymph nodes, these roundish yellow structures in different parts of the body. They can filter the lymph for us. We have about 600 lymph nodes in all parts of our body, but they are more accumulated in the inguinal region, intestinal region, mammary region, the axillary region, and also our cervical region. In these parts, the accumulation of the lymph nodes are more. In this picture, you can see the tonsils and also the pierced patch of the small intestine. So these are the overview of lymphatic structures which we have in our body. And the first structure which I want to start with is the lymph node. I told you that we have about 600 lymph nodes in our body. We can see the accumulation of the lymph node in the inguinal region or groin. We call them inguinal lymph node. We can see them near the intestine, so we call them intestinal lymph node. We have many lymph nodes associated with breast, so we call them mammary lymph node. You have them in your armpit, which we call them axillary lymph node, and also in the cervical region, which we call them cervical lymph node. But what is the structure of the lymph node and how the lymph node can filter the lymph for us? This is the macroscopic view of the lymph node. Our lymph node has different parts. As you see in this picture, we have a pink part for our lymph node, which we call it capsule of the lymph node. The capsule can cover our lymph node from outside. This is the dense irregular connective tissue. Then from the capsule, some invaginations come to the organ, and these invaginations are called septa, or we can call them trabeculae. These invaginations are short, and they can penetrate to the organ as you see in this picture. For the lymph node, we have two distinct regions. One region is located outside and one region is located in the center. The region which is located outside or periphery is called cortex, and the region which is located in the center is called medulla. In the cortex and medulla, the organization of our lymphocytes are different. As you see in this picture, in the cortex, we have lymphatic follicles. They are roundish structures. And in the medulla, we have medullary cord. They are chain-like organization of the lymphocyte. So in the cortex, we can see lymphatic follicle, while in the medulla, we have medullary cord. And we have one part in our lymph node, which we call it hilum or hilus. This part is the indented part, and through this part, we have entrance of the artery, exit of the vein, and also exit of efferent lymphatic vessels. The other parts which I want to talk about it in the lymph node is about the flow of lymph. The lymph comes to the lymph node through the capsule from the afferent lymphatic vessels. The afferent lymphatic vessels are numerous and they can bring the lymph to our lymph node. Then the lymph flow in the subcapsular sinus beneath the capsule. Then the lymph come to the trabecular sinus around the trabeculae. And after that, the lymph continue to flow in the medulla, in the medullary sinus. And at the end, the lymph come out of the lymph node through the efferent lymphatic vessel. And in this picture, you can see 1 and 2 lymphatic vessels, which plays the role of efferent. So this is the general structure which we have for each of the lymph nodes. In the next picture, in the next slide, you can see the other macroscopic structure of the lymph node. In this picture, you can see the capsule which cover the lymph node from outside. You can see septa or trabeculae, which are short and come to the lymph node. You have the cortex with roundish lymphatic follicle and the medulla with the medullary cord. You have the hilum region, the region which the blood vessel pass through it, and the efferent lymphatic vessel leave our lymph node. In this picture, you can see the subcapsular sinus, the trabecular sinus, the medullary sinus, and then the efferent lymphatic vessels. This is the macroscopic structure of a lymph node. This is the histology of the lymph node. In this picture, you can see everything which I mentioned them before for you. In this picture, you can see the capsule which cover the lymph node from outside. And you have one region which we call it hilum or hilus. You can see the trabeculae which come to the organ and they are short as you see here. You have 2 distinct regions. The first part, the outer part is called cortex, which you have it here. And the inner part is called medulla. In the cortex, you can see lymphatic follicles. And in the medulla, you have medullary cords. And you can see the hilum here. And if we want to talk about the flow of lymph, you have the flow of lymph like this. The lymph comes by afferent lymphatic vessel. It goes to the subcapsular sinus, then trabecular sinus, then medullary sinus, and then it comes out by the efferent lymphatic vessel. So pay attention. If you want to identify the lymph node, you need to know that your lymph node is like bean or a kidney shape. Your lymphatic follicles are located in the cortex or they're located around, and in the medulla you only have medullary cord and medullary sinuses. This is the cartoon of the lymph node. This is the other histology picture of the lymph node. In this picture, you can see the capsule, the short trabeculae, the outer part, which is full of lymphatic follicle and its cortex, the inner part with medullary cords and medullary sinuses. This is the structure of a lymph node. The next structure is the spleen. I told you that the spleen is located in the left upper quadrant in our abdominal cavity. On the surface of the spleen, we have some depressions. One depression is called gastric impression. This is the location of our stomach. And one impression is called renal impression, which is actually the location of our kidney. And here you can see the hilum of the spleen. The spleen can filter our blood. The blood can enter the spleen through the splenic artery and leave the spleen by the splenic vein. In the spleen, we have a capsule which covers the organ from outside, and you can see it by blue color here. From the capsule, we have some trabeculae, short trabeculae, which come to our organ. In the spleen, we have many lymphatic follicles, which are distributed everywhere in our spleen. It's different from the lymph node because in the lymph node, the lymphatic follicles are only located in the cortex or in the periphery. But in the spleen, they are distributed everywhere. In the periphery, in the center, everywhere. In the fresh spleen, the lymphatic follicles are grayish-white. Because of that, we call them white pulp here. In the white pulp, we have lymphocyte, B and T lymphocyte. We have macrophages, and we have a section of the blood vessels, which we call them central artery. The other parts of the spleen is red. We call it red pulp. Red pulp is actually the blood sinusoids and the splenic cord. The blood sinusoids are capillary type 3. In this type of capillary, we have large openings in the endothelial cells, and we don't have complete basement membrane. So the blood cells can easily come out of these capillaries or go into these capillaries easily. And we call it red pulp. And the red pulp, as you see here, is actually the background of our spleen. This is the other picture of the spleen. In this picture, you can see the capsule which covers your spleen from outside. You can see the trabeculae which come to the spleen, and the trabecula are short. You can see the roundish organization of the lymphocytes, which we call them white pulp. They are lymphatic nodule or lymphatic follicle, and they are located everywhere in our spleen. And we have a key for identification of them. All of them contain central artery, a branch of artery, which we call them central artery. The name is central, but actually it's not located in the center. It is eccentric. And besides the white pulp, which is dark blue in the color in our histology slides, we have a red pulp, which is the background in your spleen. It's full of blood sinusoids and splenic cords. This is the other histology picture of the spleen. You can see the capsule. In this picture, we don't have trabeculi, and you have two parts. One part are these roundish structure. They are white pulp, and your background is called red pulp. This is the other picture of the spleen. In this picture, you can see the capsule, the trabeculae which come to the organ, and you have these roundish white pulps with their central artery, and the background, which we call it red pulp. So this is a spleen. The next structure, the next organ is called tonsil. You know that the tonsil is partially encapsulated lymphatic organ. Our tonsils are located in our pharynx. This tube, which is located in the back of our nasal cavity and the oral cavity, is called pharynx. And you will learn about the different parts of pharynx in the respiratory system. We have three major tonsils. One of them is called pharyngeal tonsil in the roof of our pharynx. In the sagittal section, you can see it here. In the anterior view, you can see the pharyngeal tonsil here. It's only one. The other tonsil is paired, and we call it palatine tonsil. You can see it at the back of your oral cavity, and it's paired, one in right and one in left side. The other tonsil is called lingual tonsil. It's many small tonsils, and they are located in the base of our tongue. So pharyngeal, palatine, and lingual. We have one minor tonsil, which we call it tubal tonsil, and it's located in the opening of auditory tube or eustachian tube. I will talk about the auditory tube in the respiratory system for you. If I want to talk about the structure of the tonsil is partially encapsulated. It means that in one side, we have epithelium. In the other side, we have capsule. And the capsule doesn't wrap our organ from outside completely. In our tonsil, the epithelium can make some invaginations inside, and these invaginations are called tonsillar crypt. And around the invagination, you can see the roundish structures, which are lymphatic follicles. This is the structure of the tonsil. If I want to draw the tonsil for you, I will draw it like this. Here you can see the epithelium, which invaginate to the organ and make tonsillar crypt. So the yellow part is your epithelium. You can see lymphatic follicle around the crypt. And on the other side, you have the capsule. This is the cartoon of the tonsil. And if you pay attention here in this picture, you can see the capsule. This region is the epithelium, and it invaginates inside and makes tonsillar crypt for you. Around the crypt, you have many lymphatic follicles. In the next picture, Again, you can see the epithelium, number 2. This epithelium come down and make tonsillar crypt. You have number 3 as the capsule, and here number 6 are lymphatic follicles around the crypt. This is tonsil. And the last organ which I want to show you is the thymus. The thymus is located in our mediastinal cavity. I told you that the thymus is large in child and it's become smaller in adult. And the regression of thymus happen during our life. From the time of our birth to age 35, 45 years old, each year we have a regression of the thymus about 3%. And after 45 years old, each year we lose 1% of our thymus. So this process of regression happens throughout our life. The thymus has a capsule which covers the organ from outside. We have trabeculi which come to the organ. The trabeculi are long and they join together. When they join together, they can make a small compartments which we call them thymic lobule. In each thymic lobule, you have a region which we call it cortex. The cortex is located around. It's full of lymphoblasts, and the medulla is lighter in the color. It's located in the center, and the number of lymphocytes are few. The lymphoblasts come to the cortex, and they start to become mature. If they become mature, they come to the medulla. If they can't mature, they die in the cortex. And about 2% of the lymphoblasts become mature and enter the medulla. After the medulla, they can go to the lymphatic structures and start to defend our body. In the medulla of the thymus, we have some roundish structures, which we call them Hassel's corpuscle or thymic corpuscle. They are actually the graveyard of epithelio-reticular cells, the dendritic cells, and the dead lymphocyte. By aging, the number of thymic corpuscle increase, and we don't know their function yet. This is the drawing of the thymus. This is the cartoon of the thymus. You can see the capsule, the trabeculi which come to the organ, and the trabeculi meet each other, make thymic lobule, and in each lobule we have cortex around and medulla in the center with fewer cells. In the medulla, you can see Hassel's corpuscle or thymic corpuscle. This is the other picture of the thymus. In this picture, you can see thymic lobule. This is the capsule. These are trabeculi, and the trabeculi come into the organ and make different lobules for us. In each lobule, you have darker cortex and lighter medulla. This is the other picture of the thymus. You can see the capsule, the trabeculi, and one thymic lobule. Outer cortex, which is dark, and inner medulla, which is lighter. And I have these 4 pictures here for you. The first one is the tonsil, because we have tonsillar crypt, epithelium in one side, and capsule in the other side. Around the crypt, you can see lymphatic follicles. The next picture is the thymus, because you have compartments, you have a lobule, and in each lobule, you have darker part, which is cortex, lighter part, which is medulla. The next picture is the picture of the spleen. In the spleen, you have capsule around the organ. You can see white pulps everywhere, and the red pulp as the background. And the last picture is the picture of the lymph node. You have lymphatic follicle only in the cortex and medullary cords in the medulla. So in this slide, you can see 4 lymphatic organs together. In this picture, you can see the location of the spleen. And you see that the spleen has a hilum. A splenic artery come to the spleen from the hilum, and the splenic vein go out of the spleen. And pay attention, the spleen can filter the blood from the old and damaged red blood cell and platelets. And actually, it can participate in our immunity too. In these 2 models, again, you can see the spleen. This is the spleen in the first model, and this is the spleen in the other model which we had in the lab. You can see the splenic artery and the splenic vein, and in this model, again, a splenic artery and the splenic vein. Here is the spleen in the cadaver. You can see the structure. You can see the hilum, the splenic artery, and the splenic vein. The spleen is located beneath the diaphragm in the left side of our abdomen. Here is the histology of the spleen. In this histology, you can see the capsule which cover your organ from outside. You can see the trabeculi which are short and enter the organ. The capsule and trabeculi can make the framework of your organ. You can see these dark roundish structures which are called white pulp. As I told you before, they are lymphatic follicles. And the background is called red pulp. The majority of the spleen is composed of red pulp. This part can filter the blood cells for us, but the lymphatic follicle can clean our blood from our pathogens. This is the other picture of the spleen. You can see the capsule, the trabeculi which come inside. All of these roundish structures are white pulp, and the background is red pulp. The next structure is the tonsil. I told you that we have 3 tonsils in our pharynx, 3 major tonsils. You can see the pharyngeal tonsil in the roof of the pharynx. You can see the palatine tonsil in the back of our oral cavity, and you can see the lingual tonsil in the root of tongue. The tonsils are full of lymphocytes and can help us defend against all inhaled and ingested microorganisms. In the cadaver, you can see the pharyngeal tonsil here and the lingual tonsil in the base of the tongue. In this picture, we can see the palatine. Here is the microscopic view of the tonsil. I told you that it's partially encapsulated, so you can see the epithelium in one side and the capsule in the other side. This region is the region of the capsule. You can see tonsillar crypts, and in the tonsillar crypts, the pathogens are trapped. And around the crypt, you have many roundish structures which are lymphatic follicles. The other tonsil. Here you can see the epithelium, you can see the crypt, you can see the incomplete capsule. around the tonsil and many lymphatic follicles around the crypt. Here you can see the lymph nodes, and this picture shows you the inguinal lymph node in our groin. There are many, as you see here. Each lymph node has afferent lymphatic vessel which enter to the lymph node. Here we have 3 afferent lymphatic vessels, and the efferent lymphatic vessel, which is only 1 or 2, and leave our lymph node. So the function of lymph node is filter the lymph for us. This is the axillary lymph node. We can see them in the armpit. These lymph nodes are embedded in the adipose tissue. Here you can see the microscopic view of the lymph node. You can see the capsule of the lymph node, the trabeculae which come inside, the outer cortex, and the inner medulla. This yellow arrow shows you the capsule. The yellow arrow in this picture shows you the cortex, and also you can see subcapsular sinus beneath the capsule and trabecular sinus around the trabeculae. In this region, the yellow arrow shows you the medulla. You can see medullary cord and medullary sinuses between them. This is the other picture of the lymph node. You can easily identify it. You have cortex around and medulla in the center. In the cortex, you can see lymphatic follicles. And in the medulla, we have medullary cords. The other histology picture, you have capsule, trabeculae. This region is the cortex. This region is medulla. In the cortex, you have lymphatic follicle. In the medulla, you have medullary cords. Here you can see subcapsular sinus, then trabecular sinus, medullary sinus, and this region is the hilum and the efferent lymphatic vessel take the lymph out. And the last organ is the thymus. You can see the thymus here, right and left lobe, in the thoracic cavity in the mediastinum. And here you can see the lungs and the heart as well. The thymus is encapsulated completely. The trabeculae come inside and makes thymic lobule, as you see here. In each lobule, you have darker cortex and lighter medulla. Here you can see the remnant of thymus in adults. You see that it's become very, very small. And this is the histology of adult thymus. You see that the functional part regressed, and we have many adipose tissue which replace the functional